In the last part, we ended by talking about some very common histological stains that were used to enhance the visibility of intracellular structures. In this section, we are going to look at a very powerful variation on cell staining. It is used very often in cell biology and microscopy, and it is fluorescent cell staining and fluorescence microscopy. So, fluorescence microscopy relies on the fluorescent molecules, sometimes called fluorophores or fluorochromes. Unique to fluorescent molecules is that they absorb light at one wavelength and emit light at a specific longer wavelength. A common example is a fluorescent, which is a fluorophore, which is very common. It is excited at 592 nanometers, which is in the blue range of the light, and its emission is at 525 nanometer, which is bright green in color. So let's first take a look at a very simple diagram of fluorescence microscopy, and then we'll go into the detail later. There's a cell stained with the fluorescent, and the light for the stain is applied, which is 492 nanometer. Then all of the stain will fluoresce, emit light at 525 nanometer, which is a bright green range. But there is a problem. Not only does the fluorescent stain emit light at 525 nanometers, but there are many proteins in the cell which are autofluorescent. They fluoresce by themselves at specific wavelength, and they will fluoresce at even 492 nanometers. So they emit light at all different types of wavelengths and therefore produce confusing result. So there is a way to solve the problem before the light arrives at the detector, which is a computer screen, a camera, or just your eye, must pass through a filter. So the filter blocks all the light except the emission wavelength that you are interested in. So in the example, the filter will block all the light except the 525 nanometers, resulting in an image which is a completely black background with bright green stained objects, which is a fluorescent stain. So on the side of the comparison of a light microscope and the bright field fluorescence microscope, so looking at the fluorescence microscope, so that all the light actually comes from a lamp, and the light is focused onto the specimen through the condenser and passes through the specimen, goes through the projector lens to the detector. For the fluorescence microscope, the light source is usually a laser. The light passes through an excitation filter and only allows the light of the excitation wavelength you are interested in to pass through the filter. So if you are using a fluorescent, then the filter will only allow the wavelength of 492 nanometer to pass through. It is then reflected back by some mirrors and focused on the specimen. The fluorescent in the specimen becomes excited and emits light at 525 nanometer, and this light gets passed through a dichroic mirror which serves as a filter and only emits light at 525 nanometer, and then passes through a projector lens to the detector, which is a computer screen or simple fluorescence microscopy, your eyepiece. Now remember that here the the color of these lights, which are red and green, don't corresponding to 492 or 525 nanometer. So don't confuse with that. This is just that the green light that is going onto the sample is the excitation wavelength, and the red is emission wavelength. So here is an example of fluorescence microscopy by direct staining technique. A live cultured bovine pulmonary artery endothelial cell was stained with a green fluorescent dye that is specifically bound to the mitochondria, and the red fluorescent dye that is specifically incorporated into lysosomes, showing the localization of two organelles in the cell. There are many organelles and components that specific dyes are available, a very powerful technique and indeed a drastic improvement from the conventional light microscopy. Just keep in mind that when we are actually observing these images or taking a picture of these images, we can only see one color at a time. We cannot see the mitochondria as green and red lysosome at the same time. So the picture is first taken only with the green mitochondria. The same cell, by changing the filter of the emission wavelength, we can take a picture of the red lysosomes, and then we actually merge those two images by using an electronic merge through the computer. So you can only visualize just only one color, which the emission filter will allow to pass through. So there are some very interesting and powerful variations on fluorescence microscopy. I want to talk about three of them. The first variation is a powerful use of ion-sensitive fluorescent dyes. These dyes fluoresce in different wavelengths depending upon the concentration of the ions. So an example of an ion-sensitive fluorescence dye is a Fura-2. So it is sensitive to calcium concentration in its surroundings. So when the calcium is low, the dye fluoresce in a blue range. They fluoresce in the blue range. When the calcium concentration is in the mid-range, they are kind of a green. And when the calcium concentration is a high, this dye is usually yellow, or some cases orange or red, just like here. So this is an example of a migrating white blood cell which is stained with a Fura-2. The cell is motile and moving upward, and the calcium concentration is low as indicated by the blue fluorescence. Then the cell gets a signal to change directions and move to the right here. Can see the calcium concentration has gone up, indicating by the yellow fluorescence. So the release of calcium into the cytoplasm plays a role in directing the motile cell, which we will talk about later when we discuss the cytoskeleton and signal transduction and how calcium does that. For now, I hope you can appreciate that how ion-sensitive fluorescent dyes can provide a lot of information about the roles that ions play in the cellular functions. So the second variation of the immunofluorescence microscopy, this type relies on the fact that you have an antibody that binds to the specific protein of interest. So the immunofluorescence microscopy can achieve that you can localize individual proteins within the cell, and is a major improvement over the standard light microscopy. So to identify a certain protein in your cell, you need a specific antibody that will bind only to those proteins. So say you want to see a subcellular localization of a protein that is called the protein X. The first step is to inject the protein into the rabbit, which is foreign in the rabbit or antigenic. You have to give some booster dose, maybe 2 or 3, and wait about 8 to 10 weeks. And then you check the serum, and it should contain the antibodies of the protein. Then you humanly bleed the animal and you isolate the serum. And this antibody from which you isolated from the serum binds specifically to the protein X. The antibody may need to be purified further for better specificity. However, these procedures are fairly routine. And you don't have to use just rabbits. You can use mice, rats, chicken, goats, even llamas. There are commercially available antibodies from llamas, which are very good for producing specific antibodies, and they generate quite a lot because they are bigger animals. Just for your convenience, this inset actually do show a typical antibody. The FAB region is variable, and the tip of the FAB region in the red binds to the epitopes of the antigen and potentially could bind to any antigen epitope having billions of variations. So literally, for any antigen that you can encounter in your lifetime, there should be a binding site on this antigen binding region of some antibody. So on the other hand, the FC region is a constant region, and this FC region is constant among all the different isotypes of antibody. So what is an isotype? First of all, this molecule antibody is an immunoglobulin molecule. It's an immunoglobulin molecule which actually is made up of 4 different proteins. You have a light chain, you have a heavy chain, 2 different types. And we are not going into the detail of that, light and heavy chains. But it's an immunoglobulin, and depending upon their substructure, they actually are divided into several isotypes. The most common one is known as an immunoglobulin G or IgG. That is the most produced antibody that is found in the serum due to an injection in that. So think about that. This is an IgG from the rabbit. So all the FC portion, remember, all the FC portion for all the IgGs from the rabbit specific species, they will all be the same. So this is completely constant, whereas different antibodies will vary in their epitope binding sites. So this is the variable region. It's a constant region. And this is needed for the specificity that you need a secondary antibody to bind here. And that's why you need the specificity of this FC region. So we're going to talk about that detail in a second. So you have the antibody, and now you are ready to carry out the immunofluorescence microscopy. So the cell of interest and the red dots right here are represented by the protein X. So the first step in immunofluorescence microscopy is to fix or permeabilize the cells. Remember, the cells are in a dynamic state, so to get really a good and accurate image, you need to freeze them in time. And also, the antibodies are very large molecules. They cannot pass through the plasma membrane until and unless you actually make bigger holes and pores and make the plasma membrane permeable to these proteins. If you are using individual cells, then you can move to the next step. But if you're using a tissue, you will need to section the specimen. Then add antibody that binds specifically to the protein of your interest, or protein X that you have just raised in the previous slide. That is just called the primary antibody. And since the specimen is permeabilized, the antibody will pass through the plasma membrane and interact directly with the protein X. The next step, you are going to add a secondary antibody. It has two unique features. Number one is that the secondary antibody will bind specifically to the Fc portion of the primary antibody. And remember that Fc portion just right here, what you are seeing, that Fc portion right here, this is the Fc portion, the stem of the Y, and that is constant. So every specific antibodies against the Fc portion of a rabbit will recognize this portion. So you can purchase these secondary antibodies that are specific for the Fc region for your antibody and routinely commercially available. The second unique feature of the secondary antibody is that it is covalently conjugated to a fluorophore. And this is a chemical reaction that it was done in the laboratory. And say, for example, fluorescein. So add the secondary antibody and it binds to the primary antibody, which is binding to protein X, and it will light up those proteins in the cell, and then you can visualize the cells or the tissue specimen with the fluorescence microscopy. Now the question arises that why you actually use the secondary antibody. There are two reasons. The first, when you conjugate a fluorophore such as fluorescent twin antibody, you destroy it by denaturing a large portion of the antibody. There are huge losses. So if you take your primary antibody and you took 8 weeks or so to produce in a rabbit and was an expensive endeavor, you add a fluorophore directly to it and it will destroy around half of the primary antibody. It is possible to do immunofluorescence by that way, but it's a huge loss of the primary antibody. The second reason for using the secondary antibody is the signal amplification. So the important part is that the signal amplification, the secondary antibodies doesn't need to bind to just only one epitope of the Fc region. There are multiple epitopes of the Fc region, multiple epitopes, many binding sites, and there will be, although in these pictures just only one antibody has been shown, but multiple antibodies will actually bind to the Fc portion and amplify the signal. And the next one is also that in addition to the secondary antibodies are commercially available, fluorescent goat, mouse, rabbit secondary antibodies. These are called anti-goat, anti-mouse, anti-rabbit. So for example, this antibody is from this immunoglobulin or this antibody that we just talked about is raised in the rabbit. So the antibody that you are getting is an anti-rabbit antibody, which will anti-rabbit antibody. If this antibody was raised in a goat, you will need the secondary antibody as an anti-goat antibody, and so on. So it has to be that specific to the animal that you're raising in. So that is about that how you use the secondary antibody for the immunofluorescence. The signals actually can amplify by the labeling by the secondary antibody, and it shows right here. So in a step one, what you do, you chemically permeabilize the sample, incubate with a primary antibody, then specific to the antigen of interest. And here you can see the primary antibody being bound. And then you just add the secondary antibody, fluorochrome labeled. And you can see that in the same epitope, which is the Fc region, multiple antibodies are binding. So this is the signal amplification that we were talking about. And then you just mount the specimen and view under the fluorescence microscope. So here are some examples of immunofluorescence microscopy. A specific protein can be localized in a fixed tissue section by this indirect immunofluorescence microscopy. Why it is indirect? Because you use a primary antibody to be tagged to the protein, and you use a secondary antibody attached to a fluorochrome or fluorophore to visualize the protein. So that's why we call indirect fluorescence microscopy. So here it shows an yellow protein called GLUT2, a glucose transport protein. And in the basal and the lateral surface of this intestinal cell, you can see the abundance of these protein, but it is absent from this brush border, which is the top, which is the apical surface. It is closely packed microvilli on the apical surface facing this internal intestinal lumen. So you can see that the black portion is the intestinal lumen, and right here is the basement membrane. The sides are basically the lateral side of the cells. So the cells are elongated. These are called columnar cells. And right here in the middle, this is a connective tissue. So the cells, this is the base, that's the top, and these are the side. So you can see that the GLUT2 is distributed towards the basal surface and the lateral surface, but not at the apical surface. So you can easily see that on the other hand, this tissue specimen is also stained with Evans blue, which generate a non-specific red fluorescence, just to give you an idea that where the different things are. So this just gives you a powerful idea that how you can actually look at the immunofluorescence of a specific protein inside a cell, which we call a localization of a single protein. So another very powerful variation is double label fluorescence microscopy can visualize the relative distribution of two proteins. In double label immunofluorescence microscopy, each protein is labeled by a different fluorochrome to compare localization. So here, a fixed and permeabilized culture cell incubated with rhodamine labeled phalloidoin, which is red. Specifically, this binds to the actin, filamentous actin. And the rabbit antibody reacting to tubulin is a major component of the microtubules, followed by a fluorescent-labeled secondary goat anti-rabbit antibody in green. So what do we mean by this? So the primary antibody is rabbit. And the secondary antibody is goat, means it was raised in goat anti-rabbit. Just make sure that you clearly get this concept. So how this secondary antibody is raised? So any general antibody, which is an immunoglobulin from the rabbit, if it is injected in a goat, it will actually develop antibodies inside the goat against this antibody, which is your rabbit antibody. So that is called the antibody goat anti-rabbit antibody, which will bind to the Fc portion of the rabbit antibody. So here the microtubule is being used by indirect immunofluorescence, whereas the first one is actually a direct staining by phalloidoin, and which is a direct fluorochrome conjugated to the drug that binds to that. So this is a direct immunofluorescence on the other hand. But on the other hand, you can actually raise antibody against actin, and you can do the same technique by this process as well. So you can have many different variations. So you can do multiple color staining. So this type of microscopy can visualize relative distribution and abundance of two or more proteins. In the top left, say for example, we just saw the example in the schematic diagram, and this is a real picture. The top left panel shows the fluorescent stained tubulin, which forms the microtubules in the cell. The top is a rhodamine stained actin, which forms the microfilament of the cytoskeleton. You take this picture and this picture separately, and you merge them to see this. So the lower panel shows the electronically merged image. Now, please be aware that your eye or the camera can see the two or more fluorescent proteins at the same time, because you have only one emission filter in there. It will only allow to pass the green light or the red light, not both. So you have to take these pictures separately and electronically merge them. This is a very important concept for your critical thinking question, so make sure you understand this concept. In the right, you see these are the cultured nerve cells, which are specific cytokine, which is found in the cytoplasm, stained with red secondary antibody. The nuclei is stained with a dye, which is known as a host, a blue nuclear stain, and have affinity for the acidic residues or the because the nucleic acid is negatively stained. So the host dye is very specific to that nuclear stain. And the cytoskeletal components are actually stained with a green fluorescent protein. So you have a 3-color staining in this process. You can have multiple capable of even 5-color stains. There are all sophisticated microscope can do multiple of them. So the next one is that there's another variation of this process is localization of the protein with the epitope tag, where you can actually bypass the antibody raising process. So you need to do this to do this, you need to have the recombinant DNA technology. Basically, you take the gene of your interest, say suppose X, and using recombinant DNA technology, you fuse an epitope which is a short peptide sequence. And a couple of commercial names for these are like FLAG, MIC, HA. There are many of them. These are just a short peptide sequence, and there are plenty of them are available. So you know the gene sequence of that, and you actually make a recombinant, which is a chimeric gene having a gene of that epitope just at one end, which could be the N terminal of the protein or could be the C terminal of the protein. Either end of the protein, you can fuse that just to make sure that you have to do a kind of an experiment where the protein is not losing its function and it will behave naturally. Most of the time it does, because these tags are really small and really does not alter the protein function. So in a cell, this protein can be detected by a fluorophore, which is attached to an antibody. Why this is an advantage? The advantage is that suppose this is FLAG. Commercially available, these anti-FLAG antibodies are easily available. Suppose this tag is MIC. Commercially available anti-MIC antibodies are available. So all anti-epitope like anti-FLAG, anti-MIC, anti-HA, which is an influenza virus, hemagglutinin antibodies are readily available. And that's the reason that as they are commercially available and used much, the cost is extremely affordable. On the other hand, what you are doing doing this molecular biology or the recombinant DNA technology just need a couple of days. Instead of that, if you are just trying to raise a primary antibody, that will take you weeks, 8 to 10 weeks. And you can just produce this protein, recombinant protein with this tag within a week, and you can just detect them. So it drastically cuts down the time that is involved in the experiment and also cuts down the cost of the experiment. So here is another example of the immunofluorescence microscopy. Co-localization of several proteins can be studied. In this picture, this MIG and the FLAG tab were detected by their specific anti-epitope antibody conjugated to a red fluorescent dye. That is why they all appear red. This process still need to fix and permeabilize the cell, and they are not living cells. In the next section, we will describe an improvement over this process in the living cell. So the last indirect immunofluorescence technique that I have talked about is with the fixed cells. Now, the fixed cells are dead as a result of fixation, which required to permeabilize them so that large antibodies can get into the cell. At some point, you may want to follow a protein in a live cell in real time. So there is a variation on the fluorescence microscopy that looks at live cells, and you can track proteins in real time. This type of microscopy relies on a protein that was originally isolated from Aquaria, which is actually a jellyfish. And they make a protein called the green fluorescent protein, or GFP. It fluoresces green range. The excitation and the emission wavelength ranges are very similar to fluorescent, which is discussed earlier. Excitation of this is at 492 nanometer and emission is at 525 nanometer. So here you are seeing the molecular structure of the protein, and these are called beta barrels. So you can see these flat ribbon-like structures are basically beta sheets, which are crisscrossed into a barrel-shaped structure. You are seeing this barrel from the side. It is actually completely a hollow round barrel if you look at it from the top. So the main take-home message from here that the center of this, the reason it fluoresces is due to a collection of aromatic amino acids that's form a ring at the center of the protein, and the electrons actually circulate in the aromatic ring and gives the protein its fluorescent properties. Now we exploit this protein in many different ways. So what scientists actually did, there are many different color fluorescent proteins are now available, and they have been enhanced by recombinant DNA technology. These are variants on GFP, can be used for tracking different proteins. So one is the variation of the GFP is an eGFP, which is enhanced grease fluorescent protein. It is a GFP that has a point mutation. One of the amino acid has changed, and it makes the fluorescence brighter and then standard GFP. There are also similarly CFP, enhanced cyan fluorescent protein, and is a blue, and also you can have an E blue fluorescence protein. You can just have all these. And you can see that you can have a yellow fluorescence protein or a red fluorescence protein. And you can see that the scientists actually isolated a couple of them naturally, but many of them actually has been made by recombinant DNA technology. And these are engineered proteins, and they fluoresce at all different wavelengths. So their emission wavelength are at all different ways. So the proteins can be labeled and visualized in living cell over the time by using recombinant DNA technology to make the fusion proteins with naturally fluorescing proteins, such as a green fluorescent protein or any of these. Now, these tubes show the emission colors, many of these different proteins, and we are going to see a nicer picture that actually is really, really cool. That you can see that here is an agar dish. So this is a bacteria are producing all these different types of proteins, which are excited to a certain wavelength, and all of them actually do have a same excitation wavelength. And supposedly this is in the ultraviolet range. So if you actually do have an ultraviolet, if you just put this one under a black light or ultraviolet light, you can see that all these different bacteria producing these different fluorescent colored proteins are fluorescing. Now, how this observation or this picture is taken all together. So you can see that all these fluorescence proteins are actually taken together because this is very, very large, and you can visually see that they are spatially actually separated out. But this you cannot do under the light microscope when you are visualizing an individual cell, because it is so small. If you have a green or red color together or something like that, it is very confusing, and you will not have a specific filter for the emission filter, which can filter, which will allow both of them to pass. Now, you can remove the filter and you can allow all of them to pass. But at that microscopic scale, all of them will actually look a very blurred vision, and you will not get a nice picture. So in a fluorescence microscopy, in reality, you have a specific emission filter, and you will see each of these colors individually one at a time. And then if you wish, you can merge them all into one image. But that's not the case because this is very large. This is a Petri plate 10 centimeter wide. So this is large. So we talked about that how we actually make a protein which is a fusion protein with your own protein that you want to tag in a living cell. So this is a similar technique that like the epitope tagging. You use the recombinant DNA technology basically you take the gene of your protein of interest, and using the recombinant DNA technology, you attach the gene that encodes. So here, for example, it is a GFP or eGFP or yellow YFP or RFP, doesn't matter. So the GFP will fluoresce, and the excitation wavelength is applied to 492, and you should be able to track down the fluorescence of the protein in the living cell. Most of the times when you tag and make this fluorescence protein, many of the times it actually doesn't alter the native protein function. Sometimes it can, but many of the times it doesn't. So here is an example of Dictyostelium discoideum. This is a non-pathogenic amoeba expressing the GFP with a protein called RAB8. RAB8 is a protein that regulates actually vesicle trafficking, and we'll discuss more later in the course. So in the left, you can see that these are the vesicles all over the place in these cells. On the right-hand side, you can actually see a phase contrast microscope, which is a light microscopy. You can easily see that the distribution of these cells, and it shows there are some spots where it is not, these vesicles are not there, but you can easily see the information that you received from a fluorescence microscopy is many, many times useful compared to conventional bright field microscopy or polarization microscopy. So another example is of how to track the live cells. So again, these are Dictyostelium discoideum, which is a non-pathogenic amoeba cells expressing a GFP tagged version of an actin-binding protein. Now, just please remember that these pictures are right here. These were actually GFP tagged proteins. So originally, when looked or taken the picture on a colored plate, they probably will be green. So these color will be green, but this is a black and white image. What you are seeing that the bright white is the fluorescence. Please remember that. So these 4 panels are basically the time-lapse field microscopy. On the left are the fluorescence image. On the right are the corresponding bright field image. Each panel is a frame in a movie using the video microscopy. So in the fluorescent images, the actin is lit up and generally is on the periphery of the cell. The main interest that was the role of the actin in endocytosis, which is a process by which the cells take up particles from an extracellular space. To do this by forming a vesicle from the plasma membrane and then pinched off, end up in the cytoplasm. This is a cell undergoing active endocytosis. So the frame 1, red arrow right here, the red arrow, see the cell is forming an endosome and brightly fluorescing, indicating the presence of actin. In the frame 2, you can see the progression and the formation of the vesicle, and the process continues to invaginate into the plasma membrane. Frame 3, you can clearly see that the vesicle has been pinched off within the cytoplasm and surrounded by actin. Frame 4, you can barely see the vesicle, and the vesicle is still there, but there is no actin there as such. So this experiment actually shows that the actin is very important in on the formation of endosomes. Pinosomes are just endosomes. Pinosomes or pinocytosis is a type of endocytosis which pick up small molecules or liquid water droplets, whereas a phagocytosis is taking up larger particles or food. So pinocytosis and phagocytosis are just 2 types of endocytosis, but this is endocytosis as well. So don't get confused by what is meant by the pinosomes. So this is a pinosome, which is a endosome vesicle as well. So the experiment shows that the actin is very important, as I said, and you can appreciate that how powerful this GFP tagging protein and tracking the GFP of the proteins in the live cells actually do mean. So you can insert the modified GFP or any other protein in developing organism at the right time for them to be expressed in virtually every cell. So these are the transgenics and examples of zebrafish, which you can purchase probably in Walmart or some hobby stores. Also, you can have a fluorescent plant, tobacco plant under the black lights. And here you can just see that there are some recombinant, these are some transgenic mice, which will fluoresce like this under the black light or the ultraviolet light that you see. And this mice right here is non-transgenic and is not fluorescing. So this is just an idea that how powerful this technique is. So this will be the end of the part 2B, which is the fluorescence microscopy. And in the next part, we are going to talk about the different types of modified fluorescence microscopy, including confocal microscopy, 2-photon microscopy, and several others. So this is the end of part 2B, fluorescence microscopy.